Very good morning to everyone. So first of all, I would like to thank the organizer. Can you hear me? First of all, I would like to thank the organizing committee and the administration of the KSR group of institution for giving me this wonderful opportunity to present my research. So today I will be uh, discussing about the polyphenols, an effective platform in bio-nanotechnology. Um, Before going to the main presentation, let me introduce myself. I am Dr. Satish Kumar, working as a postdoctoral researcher in School of Materials and Energy, Southwest University. So this is the place where I am working. It is you can pronounce the state as Chongqing. It is located in the southwest part of the China. So you can see this city is basically constructed in a hill station. So it is a beautiful city, and you can find my university campus and my lab here. So and this is the place where the two major rivers of China will meet. This is Yangling River and Yangtze River, and this is Jiangling River. So you can see this. One side of the river is yellow in color, and another side is green in color. So the, that's all about myself. So now I'll move on to the main presentation. So I hope yesterday in the previous session, Dr. Gopala Karunakaran has spoken about the basics of nanotechnology. So I don't want to take much time in giving information, background information about nanotechnology. So I'll just go through some basic information about nanotechnology. So basically, nanotechnology involves with the design, synthesis, and manipulation of materials or atoms or molecules with size range between 1 to 100 nanometer. That is 10 to the power of minus 9 meter. It is 1 billionth of the meter. So the word nano is derived from the Greek word nanos, which means a dwarf. The term nanotechnology was coined by the Japanese scientist Norio Taniguchi in 19. So the need for nanomaterials. So silver in any form will give antibacterial property, but bulk silver will also give antibacterial property. But the need for nanomaterial is when the bulk material is converted into nanomaterial, it will give some improved physiochemical properties like surface area to volume ratio and improved optical, electrical, mechanical, and biological properties. So this is the main purpose why we are going for a nanoscale material for the fabrication of nanoscale material. So nano is not new. It is already exist in the nature, as well as you can see some of the ancient sculptures having the nanotechnology. And nano in medicine is also a new thing. It is already there in our ancient Ayurvedic system. You can see the Ayurvedic bhasmas are having nano formulations like gold nanoparticles is in Swarna bhasma, and copper nanoparticles in the another. There are plenty of bhasmas have been formulated using the nanotechnology in ancient times. So yesterday's talk, Dr. Gopal Karnavaran has spoken about the basics of nanotechnology. So today I will deal with how these nanomaterials can be synthesized and the characterization of nanomaterials. So it will be I will discuss with some technical sorts of evidence. So here, synthesis of nanomaterials is broadly categorized into two types. One is a top-down approach, and another one is bottom-up approach. In the top-down approach, the bulk material will be converted into a nanoscale form. In the bottom-up approach, small atoms will be assembled together to form a nanoparticle, to form a nanomaterial. This process is known as a nucleation process. The synthesis of nanomaterials can be achieved through different methods, physical methods, chemical methods, and biological methods. You can find different physical techniques that has been used for nanomaterial fabrication, like vapor deposition, microwave irradiation, ablation, sonochemical techniques, and here in the chemical techniques also are there. The chemical reduction method, microemulsion, thermal decomposition, electrochemical analysis. So these kind of physical and chemical techniques can be used to fabricate different types of nanomaterials. In recent times, researchers have given much attention towards the biological synthesis of nanoparticles, because in physical and chemical methods there are some limitations, there are some drawbacks are there. So now people are interested in synthesizing nanomaterials through biological means. Like in biological mediated nanomaterials, we can use different bioentities like bacteria, fungi, yeast, and actinomycetes. In case of the plant, different parts of the plants like a leaf, fruit, peel, stem, root, flower, latex. These plant entities have been used to fabricate various nanomaterials. So ultimately, these nanomaterials can be used for different applications, right from energy, environment, and healthcare applications. So our focus is towards healthcare applications of these nanomaterials. So these nanomaterials can be used for drug delivery applications, theranostic device development, hemo and photothermal therapy, antimicrobial, anti-cancerous drug development, tissue engineering and regenerative medicine, biosensors, and another different diagnostic device development. So when you are synthesizing a nanomaterial, we should focus two major things. One is reducing agent, and another one is the capping and stabilizing agent. The reducing agent will act as a reducer. It will convert the bulk material into nanoscale material via reduction process, and the capping agent will give stability. So after bulk material is converted into nanomaterial, the nanomaterial should be stable for a long time. So this capping agent will protect the nanomaterial from the aggregation, and it will also make the nanomaterial stable for a long time. So as I told, in the biological synthesis, uh, microbial mediated synthesis is also there, and plant mediated synthesis. In case of microbial synthesis, the process is quite tedious. You have to maintain the microbial culture for a long time, and it also requires some purification steps in the downstream processing. So in case of plant materials, it is a very simplistic method. You can take a plant, any of the plant part, and you can just make an extraction, and you can we can fabricate different type of nanomaterials. So our focus is also towards the plant mediated nanomaterial synthesis. So initially we have started with the screening of different plant materials, medicinally important plant materials. So in that we have found out that for plant materials, the plant polyphenolics and the reducing sugars and proteins are largely involved in the reduction process. So it is acting as a reducer. It will convert the silver ions into silver nanoform. So in that reduction process, these polyphenols, reducing sugars, and the proteins are largely involved. So that from that we have screened the different plant materials. Here, one of our study, the beginning stage, we have used this plant Moringa citrifolia. It is also known as noni. It is a very important medicinal plant. We have used this plant to synthesize silver nanoparticles. Here you can see the synthesized silver nanoparticles in the 10 to 60 nanometer range. So whenever you synthesize a nanomaterial, we have to characterize using different techniques. So the characterization study should be dependent on your nanomaterial, what kind of nanomaterial that you are fabricating. So in case of silver and gold nanomaterials, these are all some noble metal nanoparticles. It will give some optical properties. It will have some optical properties, and so that optical properties can be used to screen that particular nanomaterial. So here we have used another important medicinal plant, Dendrophthia falcata. It is a known anti-cancerous plant material, and we have used this to fabricate cellular nanoparticle towards the human breast cancer treatment. So here also you can find the synthesis nanoparticles with 5 to 45 nanometer range. So these are all some basic information that I'm giving before going to my major research work. So this is how we have why I'm giving this information. This is how our research has evolved day by day. From initially we have started with plant materials, then towards how we have moved towards polyphenol based nanomaterial engineering. So another plant material here we have used the cannonball fruit as a reducing and capping agent towards the synthesis of gold nanoparticles. Here you can see the mixture of spherical and triangle shaped gold nanoparticles with a size range of 25 nanometer. So these nanomaterials have been used for different antioxidant activities. And then we have screened another plant, the same cannonball fruit, for the synthesis of different nanomaterial. That is palladium nanoparticle. Here you can see the palladium synthesis palladium nanoparticles with the size range between 5 to 15 nanometer. So after screening so many plant extracts and plant components, we have found out that the secondary metabolites, polysaccharides, polyphenols, these are all playing a major role in the reduction process. That is converting the metal ions into metal nanoform. So these plant components are largely involved. So from that identification, we have moved on to the synthesis of flavonoid-based or polyphenol-based nanomaterials. The problem with the crude extract, when you are using a crude extract for the synthesis, it will give particles in a different size range, as well as you cannot predict the exact mechanism of nanomaterial synthesis. So to avoid this kind of limitations and hurdles, so we have focused on the polyphenol based. Instead of using a plant material, we can use you can use a particular polyphenol which is responsible for the reduction process. So as we know that there are plenty of bioactive polyphenolic compounds have been reported, and it is under clinical trial for a different disease treatment and therapeutic applications. But the successful translation of these biomolecules from preclinical trials towards the clinical trial, it is very less. So the nanomaterials and the incorporation of a nanoparticulate system can improve the bioactivities and also for the successful translation of this biocompound. So we have merged these two technologies like nanotechnology with biotechnology to form hybrid nanosystems towards better healthcare applications. As we all know that plant polyphenols are ubiquitous compounds that exist in all type of plants. So these are all some of the types of polyphenols. There is phenolic acids, flavonoids, and there are plenty of compounds. Polyphenols have been isolated, and the structural parameters have been studied. And it has various therapeutic applications like anti-cancer, antiviral, anti-tumor, antioxidant properties, anti-inflammatory, and for the wound healing and other regenerative applications. These polyphenols have been already used. So these polyphenols can be used for the engineering of different types of nanomaterials towards different applications like treatment of disease, imaging, diagnosis, and therapeutic applications. So different types of nanomaterials, right from metal oxides, 2D nanomaterial, polymeric nanomaterials can be synthesized using the polyphenols and other bio entities. So we have chosen a particular flavone, Chrysine. It is a known anti-cancer compound, which is under which has been tested with almost all type of cancer cells, and it is under a preclinical trial. So even though it has tremendous anti-cancer activity, the success rate in the clinical level is very less due to its high molecular size, poor absorption, loss of stability, bioavailability. So, in order to improve the qualities of Chrysine, we have incorporated this Chrysine in the nanosystems and towards the better cancer therapeutic applications. So we have used this particular Chrysine as I told in the previous slides. Whenever we synthesize a nanomaterial, we have to focus two things. One is a reducing agent and another one is a capping agent. Here we have used Chrysine as a both reducing as well as the capping agent to synthesize silver and gold nanoparticles. So this Chrysine will convert the silver and gold ions into silver and gold nanoforms. So whenever we are synthesizing a nanoparticle, we have to focus different parameters like the reaction conditions that we provide, right from the temperature, pH, and the concentration of reducing agent as well as the concentration of the precursor that we use, and the stoichiometric proportion of these two. These parameters play a crucial role in the size and shape of the nanoparticles. It influences the size and the shape of the nanoparticles. We have to screen through UV visible spectroscopy because whenever we synthesize a nanoparticle, we cannot go for high-end experiments like transmission electron microscope or X-ray photoelectron spectroscopy. So we will initially we will screen the size and the shape of the nanoparticle using UV visible spectroscopy. Here you can see based on the intensity of the spectrum, we can predict the size. So if it is a broad spectrum like this, we can uh, we can conclude that the particles are polydispersed, which means the size range is high, between like 5 to 100 nanometers. So in case of an intense spectrum, it clearly shows that the particles are narrow size distribution like 5 to 15 nanometer. So based on the intensity of the spectrum, so basic principle of this resonance is whenever we hit the nanomaterial, the silver and gold nanoparticles, it will emit some surface plasmon resonance. That resonance will give absorbance at a particular nanometer in the UV visible spectrum. So silver will always give absorbance at around at 420 nanometer. So based on the SPR peak, we can confirm the synthesis of particular nanoparticles. So that is silver nanoparticle. In the same way, for the synthesis of gold nanoparticle also, we have to fix all the parameters, and based on the FTR spectrum, the gold will give SPR resonance around at 530 to 560 nanometers, and based on the color observation also, we can predict the synthesis of successful synthesis of nanoparticles, gold nanoparticles. So another important characterization is FTIR, that is Fourier transform infrared spectroscopy. It is very important for to screen whether the cries the to check the role of Chrysine in the reduction and stabilization process based on the functional groups in the transmittance. We can predict the which functional group of the Chrysine, that is the hydroxyl or carbonyl group, which functional group is largely mainly participates in the reduction as well as the stabilization process. We have found out that the hydroxyl group involved in the reduction as well as the carbonyl group involved in the functionalization, that is the capping. So these two major functional groups involved in the reduction and stabilization process, that confirmation we have obtained from the FTR spectroscopy. So after synthesizing nanomaterials, we will screen that nanomaterial initially with UV visible spectrum, and then we will go for FTR spectrum. So after the physical and chemical characterization, here also this is X-ray photoelectron spectroscopy. In this experiment also, we will confirm the chemical composition of that particular nanomaterial. We will check the purity, and we can analyze the material in which oxidation state it is present, and also the capping of that chrysine on the on the surface of the nanoparticles can be analyzed through this X-ray photoelectron spectroscopy. So another important characterization is the dynamic light scattering (DLS) as well as the zeta potential analysis. Here you can see the size distribution, hydrodynamic size of the nanoparticles, and the surface charge is very important because it will show you the stability of the particular nanoparticle. If the surface charge is highly negative, so automatically if two nanoparticles comes together, due to the repulsion force, it will get repelled, and the particles will be stable for a long time. So assessing this surface charge is also very important to determine the stability of the nanoparticles. So another important characterization technique is X-ray diffraction technique. So here it will give the crystalline nature of the particular nanoparticles. As I told in the first slide, it is nanoparticles are nothing but the assemblage of atoms. So this X-ray spectroscope, X-ray diffraction pattern will give how the atoms are arranged in a particular arranged to form a particular nanoparticles. And after the spectroscopic experiments, we will screen the morphological features of the nanoparticles using different microscopic experiments, like right from atomic force microscopy. It will give the surface topology of the nanoparticles. And after the initial characterization with the morphological studies with the atomic force microscopy and field emission scanning electron microscope, you can see the particles with the size range in 7 nanometer, around 12 nanometers. You can see different sizes of nanoparticles here, silver nanoparticles, as well as the gold nanoparticles. After completing the FESM analysis to study the exact shape and high resolution morphological features of nanoparticles, we have performed a high resolution transmission electron microscope. Here you can see the nanoparticles distributed in different size range, and here you can see the arrangement of atoms in a cubic crystalline nature. This is gold nanoparticles, the HRTM image of gold nanoparticles. You can see the size distribution. So after the successful physiochemical screening, we will perform different biological applications. So before going to the biological applications, now I would like to give some information about how the polyflavonoids can be used to synthesize 2D nanomaterials. So initially I started with metal nanoparticles. So then we have screened these flavonoids and polyphenols towards the synthesis of 2D nanoparticles, 2D nanomaterials. Actually, 2D in 2D nanomaterials, the bulk materials will be arranged in a stack form. So from that, we have to exfoliate. That is, we have to peel off a single nano sheets in this 2D nanomaterial synthesis. So we will be using Chrysine again. We will be using Chrysine as a exfoliating agent to convert graphite into reduced graphene oxide. So after the conversion, here we have used also synthesized a composite of reduced graphene oxide with different metal nanoparticles. So we have exfoliated the the graphene nanosheets, and after the exfoliation, chrysin will be present on the top of the nanosheets. So we have incorporated silver nanoparticles and gold nanoparticles on that to form a nanocomposite. Again, we have used different characterization techniques right from the UV, FTIR, DLS, zeta potential analysis. We have screened. I think I have given enough information about the why we are doing this kind of experiments. So after the successful synthesis, this X-ray diffraction pattern, and after the morphological screening with the different microscopic experiments, so we have evaluated the effectivity of this particular nanocomposites as well as the nanoparticles against different cancer cells. Here we have used breast cancer cell lines, and we have initially screened with cell viability assay, and we have determined the IC50 values. So among all the nanoparticles, the silver with graphene composite has given improved anti-cancerous property, high level of anti-cancerous property. These are all some staining and microscopic experimentation that we perform to measure the anti-cancerous property of this particular nanoparticles and nanocomposites. So in case of the drug targeting, there are two different types are there. One is a passive targeting and another one is active targeting. The nanocomposites, the nanomaterials that we have formulated, it will act based on the passive targeting mode. But we can also formulate different drug delivery system for active targeting. So in case of passive targeting, the nanomaterial or the drug will be targeted based on the poor lymphatic drainage and leaky vasculature. The difference between a normal cell line and cancer cell line, the normal cell line will be tightly packed, and cancer cells will be having a poor lymphatic drainage and a leaky vasculature structure. So this will give enhanced permeability retention effect. So the nanoparticles will easily go pass through this leaky vasculature and will target to the cancer cells. In case of active targeting, we have to incorporate a ligand to bind with the receptor on the cancer cells. So in active targeting, we need all these parameters. We have to check and we have to formulate all these parameters for the successful active targeting. So this is the basic difference between drug delivery, the difference in the drug delivery applications. One is active targeting and another one is passive targeting. So apart from the cancers studies, we have also screened the nanomaterials for antibacterial applications. So here we have checked the antibacterial property of the synthesis nanomaterials against different bacterial pathogens, and we have found out that silver is giving a very good antibacterial activity against all the type of pathogens that we have tested. So here you can see as compared to the chrysin, chrysin is also having antibacterial property, but as compared to the free chrysin, the chrysin which is conjugated with the nanoparticles is giving enhanced effect, which means the incorporation of nanomaterials with the polyphenols will give us improved therapeutics. So that is the main objective of this our study, and we have succeeded in that. So these are all the some mechanistic aspects how this nanomaterial will perform against the bacterial cells, and how it will target the bacterial cell, and how it will kill the bacterial cells. So these are all some mechanistic aspects. Yeah, usually the silver and metal ions will target the cancer bacterial cells. The sulfur containing molecules of the bacterial cell like DNA proteins, and it will also uh, target the cell membrane, and it will it will lead to the cellular uptake and subsequent oxidative stress mediated bacterial cell death. So since it is having a very good antibacterial property, the silver, all the materials, we can use this materials for wound healing applications. For the disinfection of the wounds, we can use these nanomaterials. And you can see the wound contraction as compared to the positive control. Here the formulated nanomaterials also having a very good wound healing property. It promotes the collagen restoration as well as the skin regeneration process. So it is very important whenever we synthesize nanomaterial before going to the preclinical trial or clinical trials, we have to assess the toxicity of that particular nanoparticle. The nanoparticle should be biocompatible in case if you are using that nanoparticles for healthcare applications. So we'll be screening different parameters like blood parameters and histological sectioning and other parameters, the other hematological parameters to check the compatibility of these nanoparticles for clinical and healthcare applications. So here we have screened our synthesized nanoparticles uh, for their biocompatibility in a in a non-human object like here we have used a rat model. And hemocompatibility is also another parameter that we have to address before going to the clinical applications. Here you can see the nanomaterials uh, does not show any anhemolytic property as compared to the positive control, which means our synthesized nanomaterials are hemocompatible for the healthcare applications. These are all some histological uh, histological parameters that we have studied to assess the uh, toxicity of the nanoparticles. And so far I have discussed about how we can use nanoparticles for synthesis of metal nanoparticles, 2D nanomaterials with our own research outcome. So now I will discuss something about how these nanomaterials, polyphenols can be used to engineer different types of nanoparticles towards different healthcare applications. So here polyphenols was used as a exfoliating agent to exfoliate a single nanosheet from the bulk nanomaterial. It is also a 2D nanomaterial. So these polyphenols will increase the exfoliation rate uh, as compared to the other exfoliating agents. So tannic acid is a known T polyphenol, which is adhesive polyphenol. So these are all some important phytochemicals that have been used for the synthesis of various type of nanomaterials. And see, you can see apigenin, it is a known anti-cancer compound, and curcumin is also an anti-cancer compound, and catechol, it is an adhesive agent, and another naringin, and all these antioxidant flavonoids are used to synthesize different types of nanomaterials and nanocomposites. So this is an interesting topic. The polyphenol-based nanomaterials can be used for theranostic nanomaterial. Theranostic, I think you might be aware of this new technology. Theranostic, it is the combination of both therapeutic and diagnostic applications. So it will be having dual functional nanomaterials that will perform as both a therapeutic as well as diagnostic agent. So you can monitor the therapeutic response of particular nanoparticles using these kind of formulations, and it will also helps in site-specific delivery of the nanoparticles or drugs. Uh, that's all about theranostic nanomedicine, and I will tell one example formulation. Here they have used a gold nanorod in a mesoporous silica. So after loading with the doxorubicin, it is a known anti-cancer drug. It has been surface functionalized with tannic acid. It is a polyphenolic acid. So this formulation will act as a theranostic agent because it has magnetic nanoparticle as well as drug, and the polyphenols, which will give biocompatibility for the clinical applications. So the magnetic nanoparticles will give some resonance, that magnetic resonance emoji, resonance properties. So from that we can diagnose the cancer cells, as well as the therapeutic molecules will kill the cancer cells. And moreover, here we have a gold nanorod, which is a known photo absorbing agent, so that we can also perform some photothermal therapy using a laser ablation method. So these polyphenol-based nanomaterials were used in phototherapy also. Phototherapy is broadly categorized into photothermal therapy as well as photodynamic therapy. In case of photothermal therapy, we need a photo-absorbing agent, which means a nanomaterial will, particular nanomaterial will absorb the photons, incoming photons, and it will convert the light energy into heat energy. That heat will kill the cancer or bacterial cells. So based on this principle, we have exploited this photothermal therapy towards cancer treatment as well as bacterial eradication. And another type is photodynamic therapy. Here, the polyphenols like natural flavonoids like curcumin and other flavonoids were used as a photosensitizer. So when you irradiate the particular flavonoid with a particular photons, it will release free radicals as well as the singlet oxygen. These free radicals and the singlet oxygen will leads to the oxidative stress, and that will kill the cancer or bacterial cells. These are all some advanced therapeutic applications of polyphenol based nanomaterials. And here an experimental evidence that I would like to share. So here they have used a tannic acid. It is, I told, it is a T polyphenol. They have used tannic acid as an exfoliating agent to exfoliate transition metal dialkylogenides. These are all some 2D nanomaterials they have exfoliated. You can see the presence of tannic acid here. So after the exfoliations, these nanomaterial were used for the photothermal killing of bacterial pathogens. That is, they have eradicated the biofilm forming bacteria using this laser-based therapy, and they have passed the particular laser, and it will the transition metal diatogenates will emit some heat, and that heat will eradicate the biofilms. So this technology can be incorporated in regenerative medicine. For example, the biomedical implants like titanium, bone implants, or the biomedical devices can be coated with the tannic acid along with some drug molecule or bio components, so that it will protect that particular surface, implant surface, from bacterial pathogens, so that it will promote the osteogenesis process or the skin tissue regenerative process. So this the polyphenol based nanomaterials can be incorporated in the medical devices as well as biomedical implants for the successful regeneration of organs or implants. So, so far I have discussed about how polyphenols are used in the engineering of different types of nanomaterials, right from metal oxides, 2D nanomaterials, and how we can use this polyphenol as a surface coating agents as well as a therapeutic agent for the in healthcare applications. So I would like to thank my professor and my research team in China for their continuous help and support. And thank you. I'll be happy to answer all your questions one by one, one by one. I'm looking forward to your questions, please. I'm waiting for your questions. I'll be happy to answer all your questions one by one. So I got a question from Kirtana. What nanoparticles are used in insulin? Actually, uh, silica nanoparticles can be used for the insulin delivery. People use silica nanoparticles, and one interesting innovation is, based on the pH responsiveness of the silica nanoparticles, it will release the insulin at a particular pH. So this can be used as a sustained drug delivery application. So silicon nanoparticles will be effective in case of insulin delivery. Is there any nanoparticles causing mutation? Obviously, there are some toxic nanomaterials that will that may cause mutation. That is why we always focus on the toxicological implications of the nanoparticles before going to the further studies or the further preclinical studies. It is very important to study the biocompatibility as well as the toxicological implications. What is a major advantage in 2D nanoparticles? 2D nanomaterials will be having a large surface area, and the advantages of different all the nanoparticles. Different nanoparticles have different advantages. Like 2D nanoparticles can be used for the phototherapeutic applications because it is a strong photo absorbing agent. So the 2D nanomaterials and also the 2D nanomaterials will be having a rough surface, so that we can, in case of drug delivery, we can incorporate a large quantity of drug in a single 2D nanosheets. So that is the main advantage. And as I told, different nanomaterials will have different advantages. Like metal nanoparticles can be used for antibacterial, antimicrobial applications because of its lesser size. It can easily penetrate the cell membrane, and 2D nanomaterials can be used for phototherapeutic applications. So my focus is only on the healthcare applications. The 2D nanomaterials are having different applications in the energy and environmental sectors. Like in energy, 2D nanomaterials were used as supercapacitors, and in case of the environmental application, also these 2D nanomaterials were used in the degradation of different toxic components and dyes, industrial dyes. And how nanotechnology used in the treatment of cancer. I think I have given you enough information about how the nanoparticles will kill the cancer cells, and I told we can use the nanoparticles. We can, as such, as well as we can incorporate some cancer drugs with the nanoparticles for the such site-specific drug delivery applications. What are all the nanomaterials used in food preservation? In case of food preservation and food packaging, we will be using some of the nanoparticles which will be having antimicrobial property, like silver, copper. This kind of antimicrobial nanoparticles can be incorporated with different polymers to form, food, towards food packaging applications. What is proper wound healing mechanism? So we can screen the histological parameters of after creating a wound and treating with a drug. We have to check the collagen deposition and other inflammatory response of the particular drugs. We can check with histological sectioning as well as through the observation. We can come to know how the wound is getting cured. Sir, can you please tell me about how polyol method we will be using in polyol method we'll be using different reducing sugars, and in the polyol method will require heat. We have to provide uh, high temperature. That is the drawback of polyol method. But we have to use some reducing sugars along with temperature. That in that process it will give nanoparticles. You can get nanoparticles. You can go for autoclave method in polyol based synthesis. How to detect proper wound healing mechanism? I think I have answered this question. How to detect proper? We have to screen the histological parameters as well as uh, we will be using different dye-based techniques like H&E staining to screen the collagen deposition as well as the inflammatory response of particular wound, and after the treatment we'll be screening with the histological parameters. I think I have answered all your questions. Can I leave now? Okay, thank you. Hope to meet you all in the validated function. Thank you.
That was really an amazing session. Thank you, sir, for sharing your great knowledge with us in a very clear and interesting way.
ಸರ್ ಇದು ನೆಕ್ಸ್ಟ್ ಸೀಸನ್ ఉంది.
A cup of tea is an excuse to share great thoughts with the great people. Let's have a break and pull back ourselves with refreshment all over the session. I would like to request all the participants to resume by 11:30 a.m. in the respective links sharply. Thank you. Today is the day where we begin to learn, to find, and also to experience what the world is like. For the next session, we have Dr. Rajesh Pandian, who received a PhD in microbiology from Bharathidasan University, India. He joined as a research fellow and an international postdoc in Department of Civil Engineering, Yeongnam, South Korea in 2016 to 2018. At present, he is working in project titled Fabrication and Characterization in zinc, copper, and tin nanoparticles for the treatment of wastewater environment at Chennai. He has received 23 international journals. He has received 25 awards and honors, especially Outstanding Scientist Award, Best Researcher Award, and Young Scientist Award, and many more. On behalf of Department of Biotechnology Ksrcas, we are greatly honored to have Dr. Rajesh Pandian to present the fourth plenary talk in the conference. The session is over to you, sir.
Thank you for the introduction. Are you able to hear me?
Yes, sir, we can hear you, sir.
Hello. Yeah.
Sir? Yes, sir.
Yeah. Very good morning to one and all. This is Dr. Rajesh Pandian and working as an associate professor in Department of Materials Engineering and Regenerative Medicine. Once again. Yeah. This is the study I have carried out in my postdoctoral fellow in South Korea. This is an interesting topic in technology oriented and entrepreneurship oriented project. The professor who is allowed to do the photocatalytic activity of entirely different materials, which is achieving the cleaning or recovering the wastewater from the region of Daegu, which is in the South Korea. So that is the ultimate aim of this study. Or working on the technology to the society. So even though in our country also nowadays it is very prevalent, so simply they are doing their masters or PhD for their not only for their project, ultimately they are coming to the society, how it is using to the society. So that is the ultimate aim of this project. Photocatalytic activity. What is meant for photocatalytic? Photocatalytic, if the H nu, which is available in the sunlight, which is producing the heat energy to the material, and it is elevating the normal material to the excited state. So this is a ground state to excited state, elevated or triggering by the H nu. So that is the purpose of such materials. So tin oxide and titanium oxide are the major photocatalyst. So if you are producing some heat energy, it will trigger themselves. By triggering, it is producing the electrons to the system. So whatever the system, it may be a water source, or it may be a surface, or it may be a land. Okay, so this is the technology developed on 1972. Wastewater treatment has evolved significantly through the UV visible light irradiation, like many progression. That is ozonation. So ozonation, you can know from the corporation water, you can smell that ozonation or chlorination. So if you are drinking the corporation water, you can smell that chlorination, chlorinated water. So the effervescence look like that chlorinated. So that is the ultimate treatment of our corporation. Not only the chlorination, ozonation, photocatalysis, photoelectrocatalysis, carbon absorption, activated sludge process, flocculation, advanced oxidation process, and hydrogen peroxide therapy, enzymatic degradation, that is chlorination. The entire Tamil Nadu government following that chlorination, solar light, and ultrasound. So these are the progress we are following for the treatment of wastewater. Hence, the present research focuses on the environmental biotechnologies. So how we are coming to the technology to the society, how we are, your project is useful to the environment or society. So in many industrial research and development are progressing with this photocatalytic or any other different and innovative concept of the proposal. They are coming up with the entire novelty. Then, so we have to gain not only to reduce the dye degradation or else antibacterial or pure water in rainy season, we are actually heating the water resources we are consuming from wherever. It may be a groundwater or it may be a corporation water. We are boiling because of we are reducing and killing the bacterial population inside the water. So that is why we are boiling, and then we are through boiling we are reducing the dust materials also. So that dust materials are normally in nanometer in size. That is why we are approaching the metals, that is zinc, tin, titanium, like that metals with nano-sized particles. It may be attached to the surface of the nanoparticle, nano-sized bacteria, or sorry, nano-sized dust materials. In any technology oriented progression, we are using that nanomaterials. So tin oxide is connected with the or doping with the graphene oxide and CNT, that is carbon nanotubes. Bacterial and dye pollution are more major problem with wastewater treatment, and increasing number of photocatalysts are being used in industry to kill bacterial and reduce pollution. In the present study, we approach the same thing here. The synthesized nanoparticles were characterized, how the materials are organized, how it is attaching on the dust material or dye synthetic in a organ. So we are carried out that the materials is affordable and very connective to the system. So how it is reducing the pollution. So that is the aim of the study. And then the nanocomposite showed significant dose-dependent bactericidal activity. What is meant for bactericidal activity? It is reducing the bacterial population, and it is attacking the bacterial cell wall, that is rupturing the cell membrane, cytoplasmic membrane, and then it is entering into the system of that bacterial cell, and it is cleaving the nucleolus, and it is degrading the bacterial population by some analysis like disk diffusion and cell viability test, and TEM analysis and SEM analysis. It shows that the entire cleaving of the cell membrane. Okay, so So how much percentage it is having? So that is we have to calculate. Even though we are having many materials, zinc, titanium, tin. So like that, materials are having very percentile oriented degradation. So we can compare that tin having 70%, titanium having 80%, like that. So if you are having the affordable charge of the chemical that is having high efficiency of degrading the materials, at the time you can analyze that the material which is adaptable to the RO system. You know very well that RO system, that is a reverse osmosis. It is crossing a membrane. Nothing a simple concept of this RO system, crossing a membrane from one side to another compartment. So how it is crossing? While crossing, it is filtering the all dust particles and bacterial population. So here we are chosen the Daegu region that is particularly prevalence in the Escherichia coli, that is E. coli, and Pseudomonas graminis, and one more species that is uh, will be revealed in the future slides, and how it is degrading the bacterial population. And over the past decade, bioenvironmental technologies utilizing various nanomaterials have been developed to improve the physicochemical properties. So even though we are having the chemicals, but we have to improve. First, the 1972 onwards, they used some chemicals, and then they used to reduce the pollution in the pollutant in the wastewater treatment, and then they have conjugated with any other metal. So it is supporting for the high efficiency of clearing the dust particles, and then how it is reducing the bacterial population. So while conjugation, titanium plus tin having high efficiency of bactericidal activity, and then titanium having some nano absorbents. It can easily absorb the materials. So that is why another decade they have chosen double materials, that is bimetal conjugation or doping. So it is called as doping, and then also called as nanocomposite. Or else another decade they have tried trimetal, that is titanium, tin, zinc. Like that, they have enhanced their productivity by attacking the pollutant or bacteria populations. So this is the current status of my research in South Korea. The nanocomposites has a greater potential in antibacterial activity. However, antibacterial activity of nanoparticles in a significant disadvantage, which can be impacted to bacterial growth and microbial community in MBRs. What is meant for MBR? Membrane bioreactor. The entire system, it will be constituted with the building size in industrial scale. They are preparing the membrane bioreactor. It does look like a 3 floors. It's look like a wide range of, that is also known as STP, sewage treatment plant, or else WW3TP, wastewater treatment plant. So it is depend upon the industry, it may change. So the membrane bioreactor is the, so whatever the bactericidal activity it is having, so it is composed with the nanomaterials. So we are using PVDF, polyvinylidene difluoride, that is PVDF, that is a polymer which is adapting the nanomaterials, and it is killing the bacterial population and filtering the dust pollutants. Okay, so in general, antimicrobial activity is an important characteristic to kill or degrade the microorganisms or prevent from microbial growth. Various antimicrobial nanomaterials have been developed to control the microbial induced diseases. So this is the methodology we are following. Wastewater collection from the different region of Daegu, that is in the South Korea country, and then we are not focusing on the particular place. So we are randomly choosing the river water, pond, like that. So various water systems we are having. So that also we have to reduce the salinity. That is also one of the concept. So now in this research we are not focusing on that desalination. So we are removing the salt content from the wastewater. So that is known as desalination and isolation of bacterial species from the different region of the wastewater system. So identification of bacterial species through the blasting of particular microbial species and gen submission, preparation of nanocomposites. So we are preparing the different material and metals to prepare the nanocomposites. Look like already I have discussed that titanium, tin, zinc, palladium, copper. So it is very by the researchers. And then photocatalytic activity, how it is emitting the heat energy to the system, wastewater system, and how it is reducing the bacterial population and dust materials, and antibacterial properties. And the final one is publication and patenting. So this is the overall research, how the photocatalyst is reducing the bacterial population and the dust materials. So this is another diagrammatic representation, how much quantity we need for the evaluating the nanomaterials. Sorry. These are the results. Coming to the point of the results, so this is the XRD. We have chosen already said that some, we are choosing particular material, that is metals, uh, tin oxide. So this is the tin chloride as a precursor. We have to reduce that tin chloride to tin oxide. We are removing the chloride materials through the nano preparation, that is hydrothermal preparation. So it's look like a high degree of pressure to the system, that is composed with nanomaterials. So how it is reducing their size to attack the bacterial population or else any dust materials. So we have to reduce that tin chloride to tin oxide, and then carbon nanotubes, CNT, and then we used the graphene oxide. So these are the inorganic compounds locally available in any chemical industry. So that is graphene oxide and carbon nanotubes. But the tin chloride is a precursor. We are not having such a material as a individual metal, that is tin oxide. So we are reducing through the hydrothermal synthesis, high degree of pressure, that is 600 degrees Celsius. We are keeping in the muffle furnace. So muffle furnace, it reducing the metal to the 100 nanometer sized particles through the outside pressure through the heat. So heat plus pressure, an addition of pressure, it is reducing the size below the 100 nanometer. It may be a 1 to between the 100. Okay, 1 to 100. In between the nanoparticles, we are prepared, and it is adjusted with the adjunct materials, that is carbon nanotube and the graphene oxide. So two types of inorganic compounds we are having, carbon nanotubes and graphene oxide, and one more pressure we are given to the nanomaterials, that is doping materials. So one is CNT plus tin oxide, another one is GO and SNO2, and then third one is sulfonation. So sulfonation, you know very well, the sulfuric acid having more aggressive in the skin irritation while you are touching. So that aggressive property of that vulnerable property of that irritating skin allergy, look like allergy, or lesions. It will form the lesions if you touch the sulfuric acid. So that type of sulfuric acid material, it is doping with that our composite, that is carbon nanotubes, tin oxide, or else graphene oxide and tin oxide. So these metals are composed with the or blended with the sulfonation. So sulfuric acid will be treated with this nanocomposites, and we are receiving this XRD pattern. So that etching property can given to that nanocomposite through helping the nanocomposite to reduce the bacterial population in the wastewater treatment. So now you can understand what is the purpose of using the sulfonation of this graphene oxide and tin oxide and carbon nanotubes. So this is the UV pattern of the or UV absorption spectrum of the tin oxide nanocomposites. So tin oxide having the photocatalyst activity and also known as photocatalyst. So it is emitting the energy source. Through the energy source, we are absorbing that how much particles it is releasing from the nanocomposite to attack the bacterial population or dust materials. So this is the UV absorption. This is FTR absorption, how it is composed. So we have to prove in any theoretical part, we cannot tell how it is composed. So this spectrum shows that tin oxide where it is accommodated in that nanoparticle, and then carbon that is graphene oxide, nothing a simple material of carbon material, you know very well, graphene. We are using that pencil knob which is constituted with carbon. So coal, charcoal, like that. Such carbon materials, it is adapted on the nanocomposite with the tin oxide and carbon nanotubes. So in future slides, you will be understand how it is accommodating that composite. So each material will take their own part in the nanocomposite. So this is the image of tin oxide nanocomposites. So the first two slides, A and B, shows that sorry shows that the circular particles of tin oxide adapted on the particular graphene oxide sheet. So graphene oxide look like a sheet. So that sheet accommodate that nanoparticles of spherical shaped tin oxide. So first two slides shows clearly it is evidenced. We are prepared the nanocomposite. And the third and fourth slide, that is C and D figures, shows that clear evidence of carbon nanotubes. So this is look like hose pipes, so small sized hose pipes, so which is having inside a hollow space, that space accommodating that entire dust materials uh, which is available in the wastewater, so which is easily taken up the dust materials from the wastewater. So that spherical shaped tin oxide accommodate on the nanotube. So you can see the fourth figure. So the clumped nature of the nanocomposites accommodate on these carbon nanotubes. So this is on evidence for the preparation of our nanocomposite. This is another one is SEM, scanning electron microscopy image of the tin oxide nanocomposite. So here we can see the GBO, graphene oxide accommodated the tin oxide nanoparticles, and then second one is sulfonated graphene oxide tin oxide nanoparticles. The EDS shows the clear evidence of what are the materials inside: carbon, oxygen, and tin. So you can see the E and F figures, the entire chemical compounds involved in this study. So this is another one is some images of the tin oxide nanocomposite, carbon nanotubes, tin oxide, which is accommodate the carbon nanotubes with the small spherical shaped tin oxide nanoparticles. Then the sulfonated nanocomposite having the clear evidence of preparation or receiving the good quality of the materials and metals. This is application part. So how it will be useful to the industry, how the technology will be useful to the industry. So we are having two types of bacterial colonies we have received from the Daegu region, that is E. coli and Pseudomonas graminis. One more bacteria we have isolated, Pantoagglomerans, that is not given here in the study. And then how it is reducing the colony forming units, that is called CFU in a ml, a milliliter level, how it is reducing the bacterial population through the treatment of tin oxide nanocomposites. Okay, so this is the disk diffusion study for the E. coli treated with the tin oxide nanocomposite. So here you can see the reduced bacterial population in the disk diffusion test, 200 milligram per ml. Microgram per ml of concentration, that is dosage we are fixed through the previous preliminary studies, how much concentration we have to treat for the reducing the bacterial population for the wastewater treatment. So this is the disk diffusion test for Pseudomonas graminis, so treated with tin oxide nanocomposite. So the higher dose, this is called dose dependent activity. If you are giving a small dose, it will give small activity. So even though we are in any sick, like in feverish, at the time we are taking a 20 milligram of paracetamol or 40 milligram or 100 milligram. So we are taking in a different dose of concentration by the prescription with the physicians. Look like that, the bacterial population also taking up the bacteria concentration degradation through the concentration. So it is called dose dependent activity. Look like the photocatalytic activity. Now we are coming to the concept of the proposal. So already we got treated with a small materials, that is nanocomposites, tin oxide nanocomposites. That metal is emitted the heat energy through the light source. So now we are higher study to the particular system. So normally we treated the tin oxide nanocomposite to the bacterial population. In another set, we are giving some pressure from the outside source, that is light energy. We are giving the light energy, that is UV visible light irradiation. So that light source is triggering the nanocomposite materials, and it is delivering the significant activity to the bacterial population degradation. So now you can see the entire population is degrading by the time of 150 minutes. So the concentration is reducing by the 30 minutes interval. So at final 150 minutes, the sulfonated carbon nanotubes, tin oxide nanocomposite materials is reducing the bacterial population in the wastewater treatment. So this is one of the application we are done. Then this is E. coli species, how it is reducing through the treatment of tin oxide nanocomposite. This is another one is Pseudomonas graminis treated with the tin oxide nanocomposite. So 150 minutes with the given time, it is reducing the population and giving the significant activity. Okay, this is the entire study. We are comparing with the preliminary and previous researchers has done in their own laboratory. So even though we are done many researches in the tin oxide, tin sulfonated, cobalt, titanium, you can see the preliminary studies. The 18th row, you can see the present study, how it is competing from the comparable significant activity than other studies. So how it is reducing a population. So we are given the particular bacteria E. coli and Pseudomonas graminis. So the population is reduced when compared to other materials they are used in the previous research. Okay, now I am giving the evidence of the 10 images of the bacterial population, how it is reducing their own lifespan by the treatment of tin oxide nanocomposite. While treating with the nanocomposite, the cell membrane is getting cleaved. So already I have mentioned the entire materials is degraded here in this is the E. coli Pseudomonas graminis species, how it is reducing their population by degrading their own cell material. So this is clear evidence of the cell membrane having a plasma membrane without any damage or degradation. But you can see this is the another GOSNO2 treated material is the cleavage of such cell membrane. It is totally sulfonated. It is aggressive property given the most strength to cleave the plasma membrane. And then this is also carbon nanotubes and tin oxide doped materials. And then this is Pseudomonas graminis, the club of bacterial population you can see without any damage of plasma membrane. This is another treated carbon nanotubes. Here you can see the carbon nanotubes are attacking the cell membrane. Here one of the bacteria is dead, completely degraded. So this is the concept we have created, and we have submitted patent and received the patent for this nanocomposite materials. This is the entire diagrammatic representation, overall schematic for the treatment of wastewater by this tin oxide nanocomposite. This is the cylindrical approach, that is muffle furnace. We are using this wastewater treatment for the nanocomposite preparation under 600 degrees Celsius muffle furnace. And then some images of the nanomaterials and how we are treating the visible light to the wastewater treatment. This is the evidence of such bacterial population, how it is degrading or killed by that materials. So this is the entire system we are used for this carry this research. So how it is entering this is the membrane we have prepared, PVDF and PES. PVDF, polyvinylidene difluoride membrane, and then PES, polyether sulfone. So these are the membranes having the wide range of porotic membranes, which is crossing the entire water flow and then filtering the dust materials and bacterial population. Here you can see the carbon nanotubes are accommodating the space of entire porotic membranes, that is polyvinylidene fluoride or PES, polyether sulfone. So we are checking the technology, how it is useful in the attacking the bacterial population. Here you can see the zone of inhibition is take over the position to degrade the bacterial population. So coming to the conclusion, so hydrothermal method, it is a wonderful technology to preparation of nanoparticles and nanomaterials or nanocomposite. Then, so how we can use this EDX SEM images to cornering the bacterial degradation, and then what are the bacterial population is more prevalent in the country, we can choose that bacterial population for the treatment of wastewater management. So this is another 120 minutes is enough for the treatment for the membrane bioreactor MBR system for degrading the bacterial population. So these results will be useful in the future aspects of wastewater treatment involving the irradiation of bacteria. So this is the publication of myself, Journal of Photochemistry and Photobiology, Impact of 6 in 6, and then Ecotoxicology and Environmental Safety. This is another publication, Impact of 6, and then this is another publication from Korea, Chemical Engineering Research and Design, Impact of 3.7. This is final one is my major achievement in my lifetime, 10 impact of Journal of Hazardous Material for the degradation of dye. So this is my professor. I thank him for the carrier for carrying this entire research, Professor Yang Hu An. This is my team, research team. We are entirely working for the wastewater treatment. Yeah, thank you for your attention for this presentation. Thank you. Unador. Hello. Hello. Are you able to hear? Hello.
Sir, yes, sir. Hello, sir. Hello, sir.
Hello.
The session is now open for discussion. Participant can post your queries in the chat box. Thank you.
Hello, everyone.
Sir, yes, sir. Sir, I think they are typing their questions, sir. We'll wait for some time.
Okay. Yeah. Thank you.
Yes, sir. Participants, if you have any queries, please post it in the comment box soon.
Yeah, effectivity of polyphenol in seen in which samples, sir? Effectivity of polyphenol is seen. Polyphenol, it's a polymer. How we can difference this? Yeah, first of all, I will come to the first question, that is effectivity of polyphenol is seen in which sample. I cannot understand. You are using such polymers, that is polyphenols. Uh, we can take from the industry for preparing the membranes, that is ultra filtration membrane, that is known as UF membranes, or else hollow tubes look like hose pipes, hollow tubes you can prepare, but we cannot see in the sample. I cannot understand the question, Mr. Srinivasan. I cannot understand the question actually. Okay, I'm skipping to Satya. Yes, how we can difference the nanoparticles from one plant to another plant? One plant to another plant. You are synthesizing the sugar nanoparticles, right, Satya? Liquid or solid state? Hazard treatment. Srinivasan, it is a solid state polyphenols. We can prepare the membranes, ultrafiltration membranes, and then you can use in the membrane bioreactor for the treatment of wastewater in the STP, sewage treatment plant. But it cannot be cleaved from the system. It cannot be take the over advantage to the system. That is, whatever the water it is we are giving in the feed tank, the same water will be released without any mixing their own polyphenols into the system. So actually, solid state polyphenols are not cleaved from the system, and it is not contaminating the wastewater or sorry, water system. Okay, are you able to understand what I'm telling? It is a solid state, and it cannot be contaminate the water system. It is just protecting the water from the bacterial population. Okay, okay, Srinivasan, right? How we can differentiate this simple nanoparticles from one plant to another? This question also very critical. You are preparing from the plant materials, or you are telling about that STP, sewage treatment plant? Satya.
Sir, I think she is offline, sir.
Okay.
Okay, sir. Sir, we close the session, sir.
Yeah.
Thank you for your enlightening speech, sir.
Thank you.
Okay.
Good afternoon, everyone. Welcome to valedictory function of BEAST 2022 International Conference conducted by Ksrcas Trichangur. Welcoming is the noblest act of any function. By welcoming, we make the persons feel at home amidst us. Sunshine is a welcome thing. It brings brightness. May I now invite our department's sunshine, Mr. M. Prasanna Rajesh Kumar, Head and Assistant Professor, Department of Biotechnology, Ksrcas, to the welcome and gathering. Sir, please.
Thank you for your introduction. It's my pleasure in welcoming all those personalities in this valedictory functions. First and foremost, I would like to welcome Lion Dr. K.S. Rangaswamy, our chairman. And next, I would like to welcome Shri Srinivasan sir, vice chairman sir. And I like to welcome Srimati Kavita Srinivasan madam for this valedictory function. And also, I would like to welcome Dr. V. Radhakrishnan, Principal, KSR College of Arts and Science, and who is a moral support and a guiding spirit for us in whatever the things we do. He will be the guiding spirit. And I would like to welcome our principal, sir, for this valedictory function. And also, I would like to welcome all the resource person who has participated and delivered a valuable lecture despite of their busy schedule. And I welcome you, sir. And I would like to welcome the chairperson and juries of this conference. And I would like to welcome the organizers who have successfully completed a 2 days function, and now we are in a valedictory stage. I'd like to welcome our organizer, heads of various department, faculty members, and research scholars of various colleges and students, and my dear participant, to this valedictory function. And I hope this conference might have opened plenty of new avenues in this life science area. I hope we will be having a plenty of key takeaways. And this kind of activities are must for young budding scientists as well as students who always require new terms and terminologies to learn always. And I hope this conference might have served the purpose. And but not least, I would like to once again welcome all those to this validatory function. Thank you.
It's deeply satisfying to win a prize in front of lot of people. Now I would like invite Mr. T. Rajendra Kumar, Head and Assistant Professor, Department of Mathematics, Ksrcas, to announce the best oral presentation. Sir, please.
Very good afternoon, one and all present here. Here, I feel it is my great honor to announce the Best Oral Presentation Award of Beast 2022. OP07 Gnanasegaran Karthigeyan and Nadesan Sudhagar, Department of Microbiology, Muttayamal College of Arts and Science, Rasipuram, Tamil Nadu. The second person, OP10, Dr. C. Nityakala, MDS, Senior Lecturer, Department of Periodics, KSR Institute of Dental Science and Research, Tiruchengodu. OP13, Ashika Patania and Jaideep Datta, School of Bioengineering and Biosciences, Lovely Professional University, Punjab. OP37, S. Bharat Kumar, Kandaswamy Kandas College, Namakkal, Tamil. And the plant biotech Hethraj College for women. Thank you.
OP77, Tangaraj Ramaswamy, Shalini Venkatesh, Sangeeta Raja, and Tajuddin Nuruddin, Department of Microbiology, Anjak College, Sivakasi. The next one to invite is Technology. Next what goes to number 9 and those
Now I welcome Dr. G. Saravanan, Head and Assistant Professor, Department of Biochemistry, Ksrcas, to announce the best oral presentation from Inter College.
Thank you, friends. It's my pleasure to announce best oral presentation of this conference, BEAST 2022. First, OP 122, S. Madhura and M. Suganya, Biochemistry, Ksrcas. OP 130, Abhinaya S. Niveta R. Department of Biotechnology, Kaveri College for Women, Trichy. OP 163, Maulusiran S. and Priyadarshini B. Department of Biotechnology, Ksrcas. OP 166, Kavyasri Priyadarshini, Department of Biotechnology, Ksrcas. OB 168, Varsini S. Achaya S. Balamurali Krishnan RS and Bhubalan, Department of Biotechnology, Ksrcas. OP 101, Janani Soumya and Dr. V.V. Satyababu Dandrau, Biochemistry, Ksrcas. OP 120B, M. Nandini and V. Abhinaya, Department of Biotechnology, Ksrcas. Congratulations. Thank you, friends. சிடி காலேஜ் வாங்கினது ஒரு டம்மிக்கு அவ்வளவு பணத்தை செலவு. ஆனா நீ வேணா பாரு பஸ்.
Thank you, sir. Now I invite Mr. M. Prasanna Rajesh Kumar, Head and Assistant Professor, Department of Biotechnology, Ksrcas, to announce the best post presentation.
Good afternoon. It's my pleasure to announce the best poster presentation award. The first place goes to PP08 Priyanka M, Department of Clinical Nutrition, Ganga Institute of Health Science, Coimbatore. And the second place goes to PP16 M Shanti Devi, Department of Clinical Nutrition, Ganga Institute of Health Science, Coimbatore. And the third place goes to PP25 Dr. S. Shanmuganathan, MDS, Department of Oral Pathology, KSR Institute of Dental Science and Research, Tiruchengood. Thank you.
Thank you, sir. Now I would like to invite Dr. G. Satish Kumar, School of Material and Energy, Southwest University, Chongqing, China, to say a few words about this conference. Sir, please.
Very good afternoon to everyone. First of all, I would like to congratulate the organizing committee for making this event a grand success. In the past 2 days, we had very interesting and enlightening sessions delivered by eminent scientists around the globe. So personally, I would like to thank the KSR group of institution as well as the Department of Biotechnology for giving me an opportunity to deliver my research and to meet you all the young minds. So I hope all the participants had a great sessions in this conference. So again, I would like to congratulate Department of Biotechnology and the organizing secretaries for making this event a grand success. Thank you.
Thank you, sir. Building the
Great. the part of the pace of the pandemic in person even will kick-started soon. As this juncture, I would like to thank each and every single persons who are directly or indirectly involved in making this conference a huge success. Thank you all. Thank you.
Thank you for your wonderful words, sir. Saying thank you is more than good manner. It is a good spiritually. Now I request Dr. S. Bhubalan, Organizing Secretary, BEAST 2022, Assistant Professor, Department of Biotechnology, Ksrcas, to deliver a vote of thanks. Sir, please.
Thank you. Good evening to everyone. Every special moment ends, and this occasion is no exception. We participate in the journey of life. Today, you will take home the memories of the best time spent in this conference. Let's take this moment and make it worthy and remembering. I feel honored and proud to have the opportunity to present a vote of thanks on this special day. Let's me, the first of all, start by giving glory to the Almighty God for making today's and yesterday occasion a resounding success. First and foremost, I thank the college management line Dr. K.S. Rangasamy Aya, founder and chairman of KSR Educational Institution, and Thiru R. Srinivasan, vice chairman, for the constant support in all aspects. Thank you, sir. And I also express my heartfelt thanks to Dr. V. Radhakrishnan, sir, principal of Ksrcas, for his motivation to take up such academic activities. We are grateful to our head Department of Biotechnology, Dr. M. Prasanna Rajesh Kumar, sir, for his word of encouragement. Thank you, sir. I, on behalf of the Department of Biotechnology and the entire BEAST crew, I thank all the speakers here for completing their important work together and sharing your findings with us today. I should mention the deep meaning of his appreciation for Dr. Vidudalai or Regina, his explanation of meeting microbes to the technology and investigation to innovation has explored new innovative ideas among participants. Thank you, sir. In addition, we are grateful to Dr. G. Karunagaran for his talk on nanotechnology for biomedical application, which gave us a complete guide on nanotechnology. Thank you, sir. I would like to express our sincere thanks to Dr. Sendil Kumar Balakrishnan for giving excellent courage to his lecture on probiotic for human health. And I would like to express my gratitude to Dr. G. Satish Kumar for providing encouragement on polyphenol, an effective platform in bio nanotechnology. Thank you, sir. And I would like to acknowledge our gratitude to Dr. Rajesh Pandian for exploring his talk on photocatalytic activity of hydrothermally synthesized nanoparticle doped ZO and CNT under visible light irradiation for water waste treatment plant. Thank you, sir. We are all inspired by your great words. I thank Dr. D. Kamalanathan, Dr. S. Abhinaya, Dr. B. Deepak, Dr. N. Gayatri, Ms. Indumadi, Dr. L. Ramkumar, Ms. K. Shanmugam, and Dr. Shanmuganathan and Dr. G. Saravanan for their heavy cooperation as a chairperson for oral and poster presentation. After this, heartfelt thanks go to all our heads of various departments and the faculty members for their valuable contribution in all our effort. Finally, I would like to take this opportunity to record my heartfelt thanks to the organizing committee members of BEAST 2022 for the right logistic support and guidance for making this conference a resounding success. My deep sense of thanks and appreciation goes to all the participants who have attended this international conference with great enthusiasm and made this event a great success. The essence of fruit is always tested in this last, and we believe that the last will be the best. I would like to thank Mr. T. Velmurugan and Mr. R. Srinivasan for their constant support and our faculty members. I would like to conclude with a quote of Swami Vivekananda. செயல் நன்று, சிந்தித்து செயலாற்றுவதே நன்று. உனது மனதை உயர்ந்த லட்சியங்களிலும் சிந்தனைகளிலும் நிரப்பு. அவற்றை ஒவ்வொரு நாளின் பகலிலும் இரவிலும் உன் முன் நிறுத்து. அதிலிருந்து நல் செயல்கள் விளையும். Last but not least, the first rule: if you are a good learner, you will be a stronger, and one day you will become a leader. Thank you, one and all.